Today we're going to talk about transcription. The idea of transcription is the process of making DNA into mRNA so proteins can be made. Before we talk more about transcription, we should talk a little bit about the central dogma of biology. In this central dogma of biology, you have DNA being transcribed into mRNA and mRNA being translated into proteins. You can think of this whole process of the central dogma of biology as writing a research paper for a class. So you can think about all the books or primary sources you read for your research as your DNA. Obviously, you're only going to take some of the material from the books, such as the quotes and evidence, and you can think of them as mRNA. And lastly, after putting together all your quotes and evidence and modifying it a bit, you get your final research paper, and this is your protein. So we will focus on the transcription part, or the process of having your books into quotes and evidence. So one thing to remember is that transcription occurs in the nucleus. So if we continue with our research paper analogy, you can say that the library where we read the books is our nucleus. So on the bottom here, I actually have a DNA strand in which we will read and do transcription. I want you to see that there is a coding strand and a template strand, and it's the template strand where transcription actually happens. The first step of transcription is the initiation. But first you have to find the promoter of your DNA. The promoter is very important as this is where transcription has to start. You can think of the promoter as the first page of a book, because when you read a book, you would obviously start on the first page of the book. In humans, the promoter is a sequence of T, A, T, A, and we call this the Tata box. So at the promoter, this is where RNA polymerase binds, and it will start transcription. If we're going to use the analogy of the book and taking out quotes and evidence, you can say that the RNA polymerase is like your pencil. Because when you take out quotes and evidence from a book, you need a pencil to write it. Note that you also need a transcription factor, which will help with the binding of the RNA polymerase to the promoter. Also note that RNA polymerase will transcribe from the 5' to the 3'. So always remember that it will always go from 5' to the 3'. The next step is elongation. So in this step, you actually start the transcription as you keep adding nucleotides from the environment. Note that you don't actually transcribe the promoter as you start right over here. When you add nucleotides, you have to use the base pairs. So thymine will base pair with adenine, guanine will base pair with cytosine. However, in transcription and in mRNA in general, you use uracil instead of thymine. And if we're going to continue to use our analogy, you can think of this as not plagiarizing. When we take out quotes and evidence from our readings, we don't copy it word by word, but instead we change a couple words so we don't get caught for plagiarism. So it's the same idea here. For using uracil instead of thymine. Again, make sure that you're transcribing from the 5' to the 3'. And if you do the transcription, you will get something like this. The last step is termination. Termination of transcription happens with termination factors. So what happens is the termination factor will bind over here, and you will have your mRNA cleave off. So note that your final mRNA product looks very similar to your coding strand of your DNA. The only difference is that instead of a thymine, you have your uracils. And like I said before, this is just to prevent plagiarism. And after you're done with your mRNA, this will leave the nucleus and will enter the ribosomes where translation will begin.